You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Thomas Bosch. He's a professor of cell and developmental biology at the University of Kiel in Germany. So, Thomas, thank you for coming.
Pleasure to talk to you. Thank you.
Yeah, it looks super interesting. We're talking about cell senescence, immune system senescence, and things like that. So tell me a little bit about your background and your current work.
Yeah, so the main interest for all my academic life is to understand how is multicellular life organized in all its complexity? How did animals evolve? Developmental mechanisms? How is health maintained in simple life forms? How similar or dissimilar is that in more complex organisms, including humans? So these are the basic questions which we started, and for asking these questions, we use a simple model animal, a freshwater polyp, for all my life, which is a classical model system in evolutionary developmental biology. It's non-senescent, it's simple, it's transparent, it's analytically very accessible, and so a beautiful system to understand these questions. And in addition, and that's important, we realized in the last 15 years that no animal, including this model animal, is alone. This animal, as all other organisms, is colonized by a specific microbiome. And so the question from the beginning became a little more complex. Not only how is multicellular life organized in the beginning of animal evolution, and how similar is that to more complex life forms, but also what role does the microbiome play in these organisms? That's one of that's the basic question I'm interested in. But there is a second access to the current research, and that is I'm in a biomedical institution, and I'm also the faculty of medical life sciences. And we realized that in the last 50 years, humans suffer from diseases in quantity, which was never seen before. And these are mostly chronic inflammatory diseases, inflammatory bowel diseases, but there's also adipositas, neurodegeneration, behavior disorders, and many. And so the question is why in the last 50 years we see this increase in this type of diseases. And there is a very strange technology which helps to linder the symptoms of these diseases. None of these diseases can be prevented, and there is no cure, but fecal microbial transplantation helps in some of the patients, in some of the diseases, not in all of them, and contributes then to lindering of the symptoms.
So what do you call the organism with its constituent microbiome? I've heard holobiont many times. Do you have a different term for it?
Not really. There are two terms which are now widely used. One is the holobiont term, and we use also meta-organisms. We distinguish a little bit, also you can think these are synonymous terms, but the meta-organism term focuses on the interactions of many different organisms within one given organism, and that's what we are interested in. We are interested in the functional analysis. So how do the different members of this meta-organism interact? What's the function of these interactions? Holobiont you can also use, but that's a term which maybe is more descriptive. So people use it this or that, and that doesn't really matter. For the reasons I have outlined, interest in basic questions: how does multicellular life function? How is health maintained over developmental time? And what is the trouble with, in our modern times, you also may say in the Anthropocene, with these numbers of diseases so much increasing, led us to found a research center, a collaborative research center, which is funded by the German Science Foundation, which is called Origin and Function of Meta-Organisms. And so our research in a team of researchers from very different fields of medical sciences, mathematical modeling, evolutionary biologists, cell biologists, we all work on different organisms to understand these basic questions.
So what are you trying to understand specifically?
So first, there's a number of surprising findings which we got in the last few years, and I may summarize what we found before I go to the challenges, and then tell you a little bit about what we are right now just facing. The most surprising finding was, here you talk to a person who knows an organism for 30 years, a simple organism, transparent, and you think this organism must be understood. Then came 15, 20 years ago, the modern sequencing machines, and so you bring this tissue to your colleague next door and put some of the tissue into the sequencer, and then the data next morning tell you there is contamination in that sequencing. Okay, then you clean your tissue better and you bring it again to your neighbor, and he runs the sequencing machine again, and then he comes back and says again there is some bacterial microbial contamination in, and so on. And so a long story very short is very simple, that there is no contamination, but every single tissue, including the one which I thought I know very well, is colonized by an invisible entity, and that is the microbiota or the microbiome, which is the sum of the genomes of the microbes. So for that reason, we consider every single organism, including humans, as holobionts or meta-organisms. And the first surprising finding was that this microbiota in our organism, which we are working on, this freshwater polyphydra, is super specific, means you have different species in the lab. They live in plastic dishes, thousands. These are lab creatures. You feed them with artificial food. You do the sequencing, and you find that every single species has its own microbiome. Then you start to think this is impossible because they all get treated the same way. And then you ask your graduate student who discovered that, Sebastian Fraune, I don't believe that. Go out in the wild, find the species again, and do the sequencing. And that's what Sebastian did. And so we confirmed that even after culturing for 20 years in plastic dishes, you maintain your specific microbiome. That was extraordinary, and that triggered us to look more deep, because if different species have their distinct microbiota, that means that the host, the animal, we do something to maintain that specific microbiota. So the question is, how do we do that? Then came another surprise. I'm interested for a long time in the evolution of the immune system and epithelial defense, and we try to understand the genes involved and so the effector molecules. But now we realize that the immune system is the crucial part which maintains the stable, beneficial microbes. You use your innate and your adaptive immune system to maintain the menagerie of beneficial microbes you have in your body, in your oral cavity, in your intestine, and anywhere. And so does every other organism. That, if you think a little bit about that, led me to rethink the evolutionary function of the immune system. If you ask somebody what's the function of the immune system, everybody will answer, well, to defend pathogens. But I think it's more than that. I think originally the immune system was invented to maintain, to establish and to maintain a healthy, beneficial microbiome. You use killer molecules like antimicrobial peptides to kill certain beneficial bacteria to maintain a stable composition of your microbes. And occasionally the same system also then attacks a pathogen, which is usually a very rare event. So that was surprising, that the immune system is actually there to maintain a stable microbiome, this specific microbiome of a given metaorganism.
Well, wait a minute. What do you mean the immune system is there to maintain the microbiome? What does that mean?
It means that we agree now that all organisms are holobionts or metaorganisms. So that means the organism, the host, which you see, must somehow is colonized by microbes, but that must somehow be stabilized. And they live together, always in this type of composition. So the question is, how is that regulated? And it turns out that the host uses an armada of different immune molecules, including antimicrobial peptides and others, to maintain that precise and specific microbiome. That's why the immune system is there, to maintain and to help to establish the specific microbiota.
So how does it? So one example of maintaining would be, let's say the microbiome of a certain creature has 10 strains of microbes in it.
Yeah.
And strain number 10 is there always in very low numbers.
Yes.
If strain number 10 got to high numbers, that would be like a dysbiosis.
Absolutely.
So you're saying that the immune system of that creature is sophisticated enough to say, all right, we want number 10 in there, for instance, but they need to be kept to this level and in check. And if that's the case, what is the mechanism by which it would accomplish that?
Yes, so it's precisely as you say. The microbiome is a complex composition of some colonizers which are more abundant than others. And the question is, how is that regulated? And we think, and we have proven that functionally by transgenic experiments, that the host has dozens, hundreds of antimicrobial peptides which function very specifically. And some of them target one microbiome group and others target another one. So, and by this cocktail of antimicrobial peptides, the host is able to maintain the specific microbiome. We proved that by knocking out, doing transgenic animals and knocking out some of these immune molecules and then exposing this host to an armada of different microbes, and we could find that the normal specific microbiome is not established in these knockdown animals. Conclusion, but because we knocked down a specific family of antimicrobial peptides, that these peptides contribute to the maintenance of this specific microbiome. Got it?
Yeah, I've got it. What do you think is informing the host, as you call it, of which antimicrobial peptides to create and in what abundance? Do you think that there's communication between the constituent microbiome? Because there would have to be a memory of this that goes transgeneration. I can't even say it. I remember this.
Yeah, excellent point. And it's not precisely understood, actually. How does the microbiome inform the host? So we think these are microbial patterns, so small molecules from the microbes, and they get received by receptors, toll-like receptors, and the toll-like receptor initiates a cascade, and downstream of this cascade are these antimicrobial peptides. So then you are back to your effector molecule, which then may again influence the composition of the microbiome. But how this precisely really works is really not yet known, as many other things in this complex metaorganism. There is a good reason why we got funded with a big center to understand precisely this type of questions. But let me.
I would think there is the host, I'm sure, has a sense of self and other, meaning its own somatic cells and then the microbiome cells. But what about the microbiome itself? Do you think that the constituent species form an idea of self and other, and that's like a collective self amongst many strains,
Okay, well, that's getting nearly a philosophical issue, but it's super interesting. I think that the microbiome is indeed a complex community which has many different members, but it has a way of communication. They do quorum sensing, they do quorum quenching, they talk to each other, and they function only as a group. We have found out, that was another surprising finding, that any function, one of the functions is, for example, pathogen protection. A germ-free animal which we can produce and maintain for a long time is highly susceptible to fungi infection. Now you bring back microbes, and then the animal is protected again. But it's not sufficient, because we have all the microbes in culture. It's not sufficient to bring one or even the dominant member of the microbiome back. And what we then say is you mono-associate the germ-free host with this type of microbe. That anti-fungi defense potential is not developed until the full microbiota is back. That means you can then give back all of the microbes, that what we call then conventionalized animals, and then protection is back. That is a very important finding, which is true we think for all other organisms as well. It's also for industrial reasons important, because it's not enough to have one so-called probiotic strains or species somewhere in culture and then try to atomize this creature and find an active molecule. This bacteria will only function in the in vivo manner when it is back in its community. So you have to somehow experimentally now make a system where the microbes have their complex, highly diverse community, plus they are living on the host and interact with the host. And only in that situation, they fully do what they normally do. And, for example, produce antifungal molecules protecting the host. That's the complexity of the microbial community, and how that complex community precisely communicates with the host is really not understood in full details.
Do you have any idea on if a foreign microbe, virus, fungus, whatever it is, attempts to invade, the holobiome?
Yes.
Where is the judging? Where is the decision making?
Yeah.
At what level is it made?
Yeah, another interesting and important question, and brings in a buzzword, and that is called colonization resistance. We think that the healthy microbiome, although we really do not know what that is, but the normal stable microbiome forms a community, and under undisturbed situation, it makes it very hard for any intruder to settle there. There can be many mechanisms and maybe several work at the same time which are responsible for that colonization resistance. That can be just physical exclusion, so there is no space where any other guy can settle, or there can be active defense. So the good guys produce something which then may hinder the intruding bacterium to colonize then the tissue there. Fact is that if we disturb the microbiome, then all of a sudden the tissue can get colonized by even pathogenic bacteria, and then causes trouble. There is beautiful work in mouse by Barbara Reherman, which shows that this function of the healthy microbiome is probably very important and is one of the features which gets lost, in laboratory animals, even if they're called wild type, and also then most likely in human populations which face Western lifestyle. And she showed that beautifully by taking real wild type mice catch somewhere in barns in the East Coast and checked the potential to prevent tumor-forming bacteria to settle the gut. And sure enough, the true wild-type microbiome has a very strong protective effect, in contrast to the microbiome which you use normally from mice, which are your control mice and are the so-called wild-type mice. Speaking to a powerful function of a diverse, healthy, wild microbiome in preventing intruders from colonizing the tissue.
When we talk about the human microbiome, it always seems to center around the gut, even though there's a vaginal one, one in the mouth, one in the skin. I mean, there's like hundreds of them.
Yes.
On the species that you looked at, or other scientists have looked at, are there microbial colonies everywhere throughout the whole organism?
Yes.
Different ones with each organ and with like everywhere in the body? Is there anywhere in an organism that there are no microbes, no potential microbial fraction?
Yeah, excellent question. We think that literally every epithelium, ectodermal, endodermal, depending on the Bauplan, on the anatomy of the animal, is colonized by microbes. The model system I'm working is nothing but a tube, and that tube is only 2-layered, and the microbes are sitting outside in a mucosa layer, and we think that during evolution, this was then an outside-inside event. So from outside, it got then into our gut-like structure, and that's why the mucosa in our case is then lining the gut tissue. But coming back to more complex animals, mammals or humans, it's absolutely true that you talked about the skin, the oral cavity, the gut, the lung, the bladder. There is, and so with more sophisticated technologies, colleagues find a stable microbiome in all of these organs. There are very few organs, if any, which are free of microbes. And comes to the critical and hotly debated issue of the uterus. We still think that the newborn baby the baby is born sterile, and then it gets its first microbes from the vagina of the mother. That's why cesarean section babies have trouble, at least statistically in terms of medical performance. So that seems an organ which is free of microbes, but the rest of the body is colonized by a complex, usually organ and epithelial-specific microbiome. Interestingly enough, they somehow also talk to each other, and the skin microbiome and the oral cavity microbiome and the gut microbiome are in exchange. We do not understand that at all, but if we remove the gut microbiome by strong antibiotic treatment or others, we find that from a completely different habitat, the oral microbes somehow are able to invade the gut and then make somehow a new microbiome then colonizing the gut, but originally now from the oral cavity. So we are really colonized by an invisible armada of microbes, and they are of essential importance for our health. Health is multi-organismic, and we know that from germ-free animals, germ-free mice and germ-free other animals, flies and worms and hydra. And of course, the effects, if you take away the microbes, are not that the animals are now completely different in their anatomy or morphology, but the effects are subtle but very important, and not so the animals don't function Normally, an evolutionary biologist would say, without microbes, you lose tremendous amount of your fitness. And there is a good reason. In nature, there is no germ-free animal. In nature, they all are colonized, and the microbes contribute to the fitness.
There's a good question. So how far down in terms of complexity of organisms do we see a holobiome or a metaorganism? Do we see it with even, bacteria themselves? Do they have constituent bacteria or viruses? Or
yes.
At what point does this stop?
Yes. Excellent question, Richard. And we think that literally there is no organism, including single cells, protists, which are in some, maybe not in all cases, associated with microbes. If you think of the origin of the very first cell, it happened that the Ur cell engulfed the bacterium, and this bacterium then became a mitochondrium, or it became, if it's getting a plant cell, became a chloroplast. And so even the getting together of the goodies of a eukaryotic cell is of symbiotic origin and is actually by engulfing other bacteria. So it goes down very much to the end of the evolutionary tree, and there are colleagues who are studying that. And the extreme form of this being together with microbes is, of course, then the microbe becomes an organelle. But in most cases, the microbes are still a living community and very active and very communicative to each other, and just living on then extracellular or in then intracellular of a given host tissue.
Yeah, there's so many questions, like I was thinking too. So let's say we have two microbial colonies inside of a creature. What defines the border between them? What's happening at the interface, let's say, of two microbial colonies? There must be a tremendous amount of information exchange to say, hey, you stay over there, I stay over here, and
yes,
interact, and we talk, and maybe we trade resources, and
it's
very odd,
Yes, see, these are all fantastic and intriguing questions, and but we still in this now emerging new and kind of really hype in the field, host microbe field, are in our early days, and in many. In few cases, we really understand functionally what's going on. You're completely right that we actually talk about that quite often, that the term boundary is getting a difficult term now, because where does a human begin or end, and where does the microbiome start? I have a perspective paper with a philosopher and a developmental biologist on the nature of self in the era of the microbiome, because previously self, what is of course something which is has to be protected, and the traditional idea was that the immune system protects the self, but now we know that the microbes are a major part of your immune system, because if you remove them, you get colonized with pathogens. So, but microbes are non-self. So non-selves protect yourself. So the immune system is not good as a hallmark of self in humans. We list two other examples. One is the brain. If you think what is you may think it's your brain. But we know, and I would like to spend there a minute just after I finish that sentence. We know that microbes interact with the neurosystem. We call it the gut microbiome brain axis, and decision making and behavior, even in simple animals which only have reflexes, is affected by the microbes. So the brain is also questionable in terms of self. Then leads as last pillar of definition of self, the genome. But we have shown several years ago that about 37% of the human genome is microbial, or can be directly traced back to the microbes. So the genome is actually a very mosaic, chimeric entity, and it just engulfed, and it got lateral gene transfer and transposons, and it got then pieces and bits from bacteria genomes. And so your genome reflects your history with the colonizing microbes. So
I've heard that endogenous retroviruses comprise, I think, like 7% of our genome, but you're saying there's also a bacterial component that comprises another 20, 30%?
Yeah. So if you can ask now your biostatistic tools, and then these are able to find then not viral but bacteria signatures in your genome, and then sure enough you will find many of them. And so this is a PNAS paper first authored by Margaret McFall in PNAS 2013, where we have a figure which shows that the human genome has 37% of bacterial traces. So all of these are exciting, but as I say, still early findings. We are far from understanding a given holobiont in all its complexity. But the most exciting development is really that we and many others found that neurons talk to microbes and microbes talk to neurons. And that if you disturb that communication between the neurosystem and the microbiota, then you end up with behavioral problems, with dysregularities, even spontaneous reactions like motility of the gut in a germ-free animal doesn't function normally. There is dysmotility, which, by the way, is one of these diseases which also is increasing tremendously. And you can rescue that by giving back the normal set of microbes. Again, not a single one, but the complex mixture. If you give it back, then this motility problem is nearly completely solved. We and others try desperately, of course, to find microbial products which are responsible for that. We are not yet there. We have to learn another language which we were not trained in. We thought we understand transcripts and genomes and peptides, but now we have to learn on small molecules. So you have to collaborate with mass spec facilities which allow you then to identify small metabolic products from microbes, which probably are the language, the communicator between the microbiota and your nerve cells. So
what if I've learned about extracellular vesicles or exosomes?
Yeah,
we know that our somatic cells produce them. I wonder if bacterial cells have their own version of exosomes. I'm sure they do that. Maybe that's the vehicle by which they communicate with our cells, and maybe they were able to interpret our cells' exosomes, for instance.
So this is another intensively investigated area right now, exosomes, which carry all kind of different content, and so bacteria also have them, and so there may be communication by fusing of these exosomes. That also may be the case. In essence, coming back to the beginning and my humble vision that I use a simple organism to understand how is multicellular life organized and how is health maintained. It turns out that we are far from really understanding that even in simple tissue, because it got so much more complicated, because there is an invisible component, which we now with modern technology, including visualization techniques, can see. And so the future must bring now a functional understanding of the interactions within such a holobiont or metaorganism, maybe with the prospect that when we understand how they communicate with each other, that we may be able to manipulate that microbiome, and that then would bring this microbiota field into the probiotic classic history. Probiotic people working with probiotics or even giving them don't know what they are doing. We have no idea how that functions, but I could see that in 5, 10, 15 years, when we really understand how that goes, that may be then a way of manipulating the microbiome. And by manipulating the microbiome, also manipulating our physiology and maybe restoring health in a time which makes it so difficult for holobiomes to maintain this diverse ecosystem.
What experiments, feasible or not feasible, do you think need to be done to elucidate some of the biggest questions that you have?
So I think we are very good in genomic analysis and so metagenomic analysis. The challenge of the day is metabolomics, so going to the metabolites and trying to understand again these complex cocktails, how are they produced at a given time during a given developmental window, and how do they then interact with host cells? Not clear at all. And the other challenge and not yet solved question is, which is very trivial, and people think we know that for a long time, but we do not know the spatial three-dimensional network and organization of these microbes in a given habitat. How do they really live together in the gut, on the skin, in the oral cavity, in a given model organism or model animal? It's completely unknown. Think about the gut, all of a mammal. All what we know about the gut is actually what we are doing, if we are honest, we sequence a little bit of poop. That means we do all the conclusions, what we have, that this person or that organism has this and this type of microbiome composition based on what comes out at the end from the anus. However, in humans, our gut is about 7 meters long, has very different habitats, and microbes live along the whole gut in more or less dense communities. We have no understanding how they really are organized. There is also a temporal factor involved. So with time, the microbial organizations seem to change. There's a brilliant researcher at Weizmann, Eran Elinav. He has published last year 2 papers in Cell addressing that question, but the technique is awful because the technique was he used human probands, and did biopsies, and then along this gut system, and then took out microbes and microbial products, and then got an idea who sits at what position, etc., etc. So in terms of the other technologies we have, that really is Stone Age tools. So what we have to develop is visualization methods to really get an understanding of the three-dimensional, the spatial organization of a holobiont. And we are far from that. To give you an example, we know there's beautiful findings from the tooth plaque. Our teeth are densely covered with the microbiome. And an American group has wonderful visualized the microbes on a given in these plaques, and they use different fluoroprobes to distinguish the different microbial partners in this plaque. And the pictures are spectacular, because what you see is a community of microbes, but if you look, which are from different members, but if you look more closely, then you find that, wow, some of them are living in downtown in this plaque, and always downtown. Others are living only in the suburbs. And there are some microbes which are using somehow, they're in between. And nobody understands what really organizes this type of the spatial organization of this given microbial community. This I consider one of the major questions which we have to solve in the forthcoming future, and brings me to another challenge, but some groups are already doing that. It's not enough that a given discipline is now engaging with high tech on that type of question. We have to combine forces. We are working inter or even transdisciplinary. And in terms of the spatial organization, we personally started to communicate with urban planners, because how a city and streets and downtown is organized. There are certain rules, there is language, there is technology involved, how to understand that. And we think we can learn from them to understand this simple but fundamental problem: how is the microbiome, the biofilm on your tooth really organized? We have no clue yet, but we have beautiful pictures which tell you that yes, there is a very high degree of organization involved.
Yeah, I mean, this is kind of maybe be a little woo-woo, but I actually see humanity recapitulating a lot of what biology has done. Like, yes. You see tiny animals with gears, and you see, like, at the microscopic level, a lot of things that people have supposedly independently invented. And, you were talking about urban planners, and our cities literally probably have a lot of the exact same interaction that microbial communities do. So,
yes,
in a weird way, it's weird. I wonder if we're being informed by our microbial constituent at all levels we don't even understand, that affects our thinking, and I wonder if we're all drawing on a reserve of knowledge that I guess is maybe invisible to us, but it's there in the biology itself.
Yes, I mean, getting a little back from but to reality, but which is also still very scary. There's absolutely plausible and reproducible experiments by colleagues in the field who transplant anxiety. And you have heard about that there's mice which you can measure if they are anxious or not by very simple experiments. So the mouse runs out of the cage, out of a black box in the cage, and is then in the open space, or is mostly in the box. And then computers and cameras can measure that, so you can quantify that. So then you identify there is a mouse which is mostly in the black box and never outside in the cage. Then you take this mouse, take the microbiome from that mouse, transplant that microbiome in a germ-free mouse, and the germ-free mouse will behave as the donor. So this is a complex behavior, anxiety, and that seems to have, maybe not all, but at least a very strong impact from the gut microbiome. And if that's the case, then you can, of course, speculate now and say, my goodness, so now when I do a certain decision, I now go in the supermarket and take that, or I do this or that. How much of that is influenced by the microbes? Interesting questions, far from being now at the moment experimentally supported, but fascinating. And again, we have to go back in biology and medicine to a holistic understanding of organisms. Previous people, my grandfather was a doctor, a countryside doctor. He looked at patients without any big technology. We have lost that. We have now all the technology. We look deep into tissue and into cells. We understand the molecular architecture of the cell completely. There's programs on that, etc. But then if you go back from this tunnel view and see the whole organism, we don't understand it, because the organism is getting diseases which we have no clue. My colleagues call them complex diseases, non-communicable diseases. So we need holistic understanding, and that's also true in biology. And only then we get a sound understanding of evolution, development, and also health and aging.
Well, very good. We've thrown around a lot of concepts, tons and tons of concepts, very unusual things.
Yes.
Perhaps to you and me, maybe because we've been thinking about these things, they're not a big deal, but to the listener, they may be like mind-blowing or revolutionary or very different. Where can they go to learn more and to take the first step on learning about what, metaorganisms and holobionts are, and, maybe read some of your papers and delve into this area?
Yeah, I mean, when people listen to this type of wonderful education programs, what you are organizing, that's, of course, one thing. One has to be careful because, as I said, there's a lot of hype in the field, and many people jump now on the train because they think they can raise interest in their things when they now use the word microbiome. So, but there are books around, small books around. Ed de Jong I am multitudes, just comes to my mind.
I remember that.
I strongly would recommend to read that. A very, 200 pages textbook. And Ed de Jong has interviewed many people in the field and did careful research on what he was writing about. So there are some books around. There's, of course, hundreds of books on the microbiome. And some of them are better than the others. So I hesitate to give recommendations. People can find information about that, but they also should be aware that, and I repeat myself now for the third time, we are in the early days. We have not yet really, we are fascinated, but we are by this complexity, but we are not yet in a functional understanding. That is also true for industry. And for using that as probiotics, industry wants to have products very quickly, but for getting there, we need a functional understanding of what's really going on. And I give you a last example, what one should not do, and that is that you take a kind of a bacterium of your choice, which you think is important. For example, lactobacilli are always important, and there are always many of the probiotics are lactobacilli. So then companies, and I'll give you a real case, but no names. Companies now think, okay, then let's now take the lactobacillus and then design and invest in a pipeline, an analytical pipeline, which is maybe millions of dollars, and then it fulfills all the safety criteria, At the very end, then you dissect these little lactobacilli bacteria into their components, and then this is followed by bioassays, cell cultures, proliferation assays, whatever, and then you have now a probiotic where you know what you're doing. That all is good and fine, but the starting point is already wrong, because when you listen to me, I told you that no bacteria works alone. And these lactobacilli in their fermenter, they do something, but they certainly do not do that what they would do in their normal community in the gut or wherever they are sitting. So the desire of developing quick and maybe cheap products in this new area of the microbiome is faced with the fact that first basic research must be done to understand what's going on.
Yeah, no, it makes sense. Well, very good. Well, Thomas, it's been a great call. I appreciate you being here, and thank you so much for your time.
Thanks, Richard. Thanks.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.